Welcome to this lecture on fundamentals of genetics. Today, we are going to explore a fascinating topic called infective heredity in Paramecium. We usually think of genetics as something strictly locked inside the nucleus of a cell, written in DNA. But what if I told you that some traits are actually infections? We will discover how a microscopic turf war challenges our basic understanding of inheritance. Let us dive in. To understand how this works, imagine you own a house and you invite a tenant to live in your living room. This tenant owns a fierce guard dog that keeps all intruders away, making your house a highly protected fortress. However, the tenant will only stay if your house has a specific type of heating system installed. If the heater breaks, the tenant leaves, the dog leaves, and your house becomes vulnerable once again. In the microscopic world, a single-celled organism called Paramecium acts just like this house. It hosts a bacterial tenant that produces a deadly toxin, but only if the paramecium has the right genetic heater to keep the bacteria alive. Now that we have our analogy, let us look at the formal biology. Infective heredity is a unique form of extrachromosomal inheritance. This means the trait is determined by an infectious or symbiotic microorganism living inside the host's cytoplasm, not the host's own nuclear DNA. Geneticist Tracy Sonneborn discovered this in certain strains of the single-celled organism Paramecium aurelia. He found that some are killer strains because they release a toxic substance called paramecin. This toxin kills other paramecia, which are naturally known as sensitive strains. The ability to produce this toxin is entirely dependent on tiny symbiotic bacteria living in the cytoplasm, known as kappa particles. This brings us to the crucial genetic catch of this relationship. The kappa particles cannot just live in any paramecium. They can only survive and multiply if the host paramecium possesses at least one dominant nuclear allele, represented by the letter K, in its nucleus. This is the heater from our analogy. If the paramecium has the homozygous recessive genotype, little k, the environment becomes inhospitable. The kappa particles will die out completely. Once they are gone, the organism permanently becomes a sensitive strain. It cannot spontaneously become a killer again just by changing its genetics. It must be physically reinfected with the particles. You might wonder why we study a microscopic turf war in a genetics class. This concept is incredibly important because it proves that inheritance is not strictly limited to the DNA found inside the nucleus. It demonstrates a beautiful, complex interaction between a host's nuclear genetics and an environmentally acquired factor. In real life, this helps scientists understand the mechanics of endosymbiosis. This is the evolutionary theory explaining how our own human cells acquired energy-producing mitochondria billions of years ago. Furthermore, it provides a foundational model for understanding how certain viruses or intracellular bacteria can be passed down from mother to offspring, affecting the traits of the next generation. To make this interaction perfectly clear, we can represent the phenotype mathematically. The formula is P equals V sub G times V sub C. Let us break down what each symbol means. P represents the final observable phenotype, where a value of 1 means the cell is a killer and 0 means it is sensitive. V sub G is the genetic viability factor. It equals 1 if the dominant allele K is present and 0 if the genotype is strictly recessive. Finally, V sub C represents the cytoplasmic state, which equals 1 if kappa particles are physically present and 0 if they are absent. Because this is a multiplication formula, if either the genetic support or the physical bacteria is missing, the product becomes 0 and the cell defaults to a sensitive state. Now that we have our formula, let us apply it to a real scenario. In our first example, a killer Paramecium with the homozygous dominant genotype, big K, and a cytoplasm full of kappa particles, mates with a sensitive Paramecium. The sensitive one has the recessive genotype, little k, and no kappa particles. They undergo a normal, brief conjugation process where only nuclear material is exchanged between the two cells. We need to determine the final phenotypes of the two X conjugates after they separate. Using our formula, we find that the first cell remains a killer with a phenotype of 1, while the second cell remains sensitive with a phenotype of 0. Let us prove exactly why this happens. Let us break down the derivation of that result, starting with the genetics. During normal conjugation, Paramecia exchange haploid micronuclei. This means they will mix their nuclear genes equally to form new diploid nuclei. Because one parent was homozygous dominant and the other was homozygous recessive, they both acquire the exact same heterozygous genotype, big K, little k. So the genetic viability factor for both cells is now 1. Both cells now have the genetic capacity to maintain kappa particles if they happen to be present. The dominant allele provides the necessary biological environment for the bacteria to survive. Moving to the next step of our proof, we must look at the cytoplasm. Because the conjugation is brief, the cells do not share any of their cytoplasmic fluid. The first X conjugant retains its original kappa particles, so its cytoplasmic value is 1. The second X conjugant receives no cytoplasm, so its value remains 0. Now we calculate the final phenotype by multiplying the genetic and cytoplasmic values. For the first cell, 1 times 1 equals 1, so it remains a killer. For the second cell, 1 times 0 equals 0. Therefore, the second cell remains sensitive despite having the correct genetics. This proves that having the dominant gene alone is not enough. The physical infectious particle must also be present. Let us look at a second example to see how changing the physical connection changes everything. Imagine the exact same two Paramecia mate once again. We have the killer with genotype big K, and kappa particles, and the sensitive with genotype little k and no kappa particles. This time, they undergo prolonged conjugation. A wide cytoplasmic bridge forms, and cytoplasm flows freely between the two cells. We want to know the resulting phenotypes after this extended meeting. Surprisingly, this time, both cells emerge as fully functional killer Paramecia, each with a phenotype value of 1. Let us derive the math behind this transformation. To prove why both become killers, we start again with the nuclear genotypes. Just as we saw in the previous example, the nuclear exchange happens in the exact same equal manner, regardless of how long the cells are connected. Both cells acquire the heterozygous genotype big K, little k. But here lies the crucial difference that defines infective heredity. Because the cells remained connected for an extended period, the kappa particles migrated across the cytoplasmic bridge into the previously empty cell. Now, the cytoplasmic state for both cells is 1, because both cells currently contain the infectious kappa particles actively swimming in their cytoplasm. Concluding our proof, we calculate the final phenotypes using our established formula. Both cells now have the required dominant gene, giving a genetic viability of 1. And thanks to the cytoplasmic bridge, both cells now have the physical kappa particles, giving a cytoplasmic state of 1. Multiplying these together, 1 times 1 equals 1 for both cells, the sensitive cell has been successfully transformed into a killer. It was literally infected with the trait through cytoplasmic transfer. And because it acquired the dominant allele to maintain it, it keeps this new killer identity permanently. To solidify this concept, let us look at this diagram comparing the two types of conjugation. On the left, you see brief conjugation. Notice the narrow connection between the two paramecia. They are exchanging genetic material, represented by the nuclei, but the green dots, which are the kappa particles, stay trapped in the original killer cell. On the right, we see prolonged conjugation. Look at the wide cytoplasmic bridge connecting the two cells. The arrows clearly show the green kappa particles flowing from the killer cell directly into the sensitive cell. This visual perfectly captures how the physical transfer of cytoplasm is the absolute key to transmitting the killer phenotype. We have reached the end of our topic, so let us summarize the key takeaways. First, infective heredity proves that some traits require a mandatory partnership between nuclear DNA and an infectious cytoplasmic agent. Second, the infectious kappa particles cannot survive without the genetic permission provided by the dominant K allele. Third, we saw mathematically that the final phenotype is the direct product of genetic viability and the cytoplasmic state. Finally, observing the difference between brief and prolonged conjugation reminds us that a cell is a complex, interconnected ecosystem. You now have a complete understanding of infective heredity in Paramecium. Great job!